Okay, so good evening, everyone. So welcome to the week 6 problem solving session of the molecular biology course. I will wait for 3-4 minutes and then I will start today's session. So I hope all of you have gone through the week 6 lectures. So in the week 6 lectures for this NPTEL course molecular biology, the concept of transcription was taught. So the topics that were covered were the transcription in prokaryotes, eukaryotes, and then followed by the gene regulation in prokaryotes. In the last session, we discussed about replication, the replication machinery, what are the enzymes involved in replication, how does the replication in prokaryotes differ from replication in eukaryotes, right? So now we are moving to the transcription process. So transcription, so in transcription, what happens? The DNA, from the DNA, the genetic information is copied into an mRNA, right? Or transcription means uh, copying of the genetic information from DNA to RNA. Now, this RNA can be mRNA, rRNA, or tRNA. Okay. Okay. So today we will cover the transcription process in both prokaryotes and eukaryotes in detail, and especially what we will do is we will solve a few sample questions, which will help you better understand the important concepts and have a quick revision as well. Will also help you to solve your assignment questions. Okay. Okay, so I will start sharing slides, today's slides. Just give me a second. Okay, so good evening, everyone. So I welcome you all to the week 6 problem solving session for this NPTEL course, Molecular Biology. This course is being run by Professor Vishal Trivedi of IIT Guwahati, and I am one of the PMRFTAs for this course. So I am Akashjyoti Pathak. I'm a PhD scholar in the Department of Biosciences and Bioengineering at IIT Bombay. So let's first discuss what all has been covered in week 6. So in week 6, uh, the topics that were covered were first in the first lecture, the transcription. We looked into transcription. We learned about the transcription in prokaryotes, right? So what is the main enzyme which is involved in transcription? And the basics of starting with the basics of transcription, what is transcription, and then we moved into how this transcription machinery takes place in is carried out in cells. So starting with initiation, so just like we had seen in replication, there were three processes: initiation, elongation, termination. Here also in transcription, there are three main processes: initiation, elongation, and termination, right? And then what is the difference between how does the transcription in prokaryote differ from that in eukaryotes, right? So basically, we talk about transcription, which means that the DNA is getting transcribed into RNA. Okay. Now in RNA, it can be either mRNA, rRNA, or tRNA. mRNA stands for messenger RNA, rRNA means ribosomal RNA, and then there is transfer RNA or tRNA, right? Now, the main enzyme which is involved in the transcription of the DNA into RNA is RNA polymerase or DNA-dependent RNA polymerase. Okay. Then, then we saw that how does transcription take place in eukaryotes, right? And then once we have the mRNA, this mRNA has to be modified, especially in case of eukaryotes. In case of eukaryotes, why the mRNA needs modification is because this mRNA, the transcription is taking place in the nucleus, while the translation will take place in the cytosol. Now, for the movement or of this mRNA from the nucleus to the cytosol, certain modifications uh, has to be made in this mRNA so that it is not degraded by ribonucleases or other enzymes, and to provide stability to the RNA molecule, because RNA is single stranded, it can easily get degraded. And also these modifications help in further translation process, which will be learning in we will be discussing in week 6. Okay. So the modification of mRNA, what are the modifications that take place in mRNA? In eukaryotes, we also learned about that concept. In prokaryotes, modification is not necessary because there is no separate compartments like nucleus or cytosol, and the transcription is coupled to translation. As soon as the mRNA is getting transcribed, at the same time, what happens? The ribosome starts binding to the newly transcribed mRNA and starts synthesizing the protein. So transcription and translation are coupled in prokaryotes because in prokaryotes there is no need of transferring the mRNA from the nucleus to the cytosol. While here in case of eukaryotes, the transcription takes place in nucleus, translation in cytosol, and there has to be modifications taking place in the mRNA to run further. Subsequent or the synthesis of the protein can take place. Okay. And then we discuss about the gene regulation. What is gene regulation? Or how is the expression of a gene regulated in prokaryotes? So we learned about the concept of operon, and in operon we looked in detail about the lac operon, TRP operon, and the ARA operon. So lac operon stands for lactose operon, tryptophan operon, and arabinose operon. So these are the different. So we will discuss this when as we go through the course. Okay. This is operon system in prokaryotes. Okay. 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 So let's start with the first question. The first question is the main enzyme required for transcription is, and the options are reverse transcriptase, DNA polymerase, RNA polymerase, restriction enzymes. Okay. So just now, as I was going through the topics that were covered, we just now discussed that the main enzyme which is involved in transcription. So it is RNA polymerase, right? So for replication, the main enzyme was DNA polymerase, and here the enzyme is RNA polymerase, or it is also called DNA-dependent RNA polymerase. Is it called DNA-dependent RNA polymerase? It is because the DNA is used as the template, and using DNA as a template, the complementary RNA sequence synthesized, right? And what are the differences between replication and transcription? So in replication, what happens? Both the strands are used as a template, and both the strands separate, and each strand acts as a template for synthesis of new daughter strand. But in case of replication, in case of transcription, what happens is only one of the strand will act as the template. Okay, so which strand is used as the template? The 3' to 5' strand is used as the template. And these are basic class 12 concepts we must have read in class 12 itself if we had biology as one of the course. So the 3' to 5' strand is taken as the template because similar to the DNA polymerase, the RNA polymerase can also synthesize in the 5' to 3' direction. It can add new ribonucleotides in the to the 3' OH group of the ribonucleotide. Okay, right. So 3' to 5' strand is called the template strand. It's called the template strand. So only 3' to 5' strand is used as the template strand. The 5' to 3' strand is not used because in that case, what would happen? That if 5' to 3' strand is also used as a template, then it would mean that the same DNA is coding for two different proteins. While we know that this should not happen, and also there can be the two strands being complementary can form a double-stranded RNA. So the only one of the strand is used as a template, and the 5' to 3' strand is called as a coding strand. Called the coding strand. Why is it called the coding strand? Because its sequence is similar, or it is same as that of the as the newly synthesized RNA. So the sequence of the newly synthesized mRNA is is similar to the to the sequence of the 5' to 3' strand. The only difference is in the uracil residues. So RNA will be having, the mRNA will be having uracil residues in place of the thymine residues. So that is the only difference. That is why it is called the coding strand. Coding strand or sense strand, and 3' to 5' strand is called the template strand or the antisense strand. Okay, so we were talking about RNA polymerase. So the RNA polymerase, as we had seen that the RNA polymerase, there is only a single RNA polymerase in case of prokaryotes. So a single RNA polymerase can synthesize all the RNA that is required for bacteria, right? And this RNA polymerase is made up of 5 subunits. It's made up of 5 subunits. It has 2 alpha subunits. and a sigma factor. So the sigma factor is responsible for recognition or for binding, or it helps the RNA polymerase to recognize the promoter region of the DNA, promoter region of the DNA. So there is a single RNA polymerase in case of prokaryotes, while eukaryotes have three different RNA polymerases, right? So they are RNA pol 1, RNA pol 2, and RNA pol 3. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. So RNA polymerase 1 transcribes for rRNA, RNA polymerase 2 transcribes for mRNA, and RNA polymerase transcribes tRNA, transfer RNA. Okay. Um, One other difference between the replication and transcription is that in replication, the entire genome is replicated, or the entire genome is copied into a copied to produce a new DNA molecule. While in transcription, only a part of the genome is transcribed. Only a part of the DNA is transcribed. So that region of the DNA which is transcribed is called the transcription unit. So the entire genome will not code for protein, or will not be is not the coding part of the genome. So most of the genome is non-coding. That means that it does not code for any mRNA. While the part of the DNA which codes for mRNA is called the coding region of the DNA. So only a part of this genome is transcribed. So that region of the DNA which is transcribed is called the transcription unit. And this transcription unit has three parts. So it will have a promoter sequence, then it will have a promoter sequence, a coding sequence, and a terminator region. So the region starting from the promoter to the terminator is called as the transcription unit. So we will look into these regions one by one. So the promoter, what is the function of promoter? What is the coding region, and what is the terminator region? Okay, the answer for this question is RNA polymerase. So RNA polymerase is the main enzyme which is required for transcription. Reverse transcriptase, if you remember, that reverse transcriptase is present in viruses, and it is also known as what? Reverse transcriptase is also known as RNA-dependent DNA polymerase, right? Because here the RNA is used as the template, and DNA is synthesized. Okay, so it is known as RNA-dependent DNA polymerase. Reverse transcriptase is for reverse transcription. That is for the synthesis of DNA from mRNA, or DNA from RNA is called reverse transcriptase is the enzyme. DNA polymerase is the enzyme which is involved in DNA replication, right? And RNA polymerase is the enzyme which is involved in transcription. Restriction enzymes are different. They are they're used in cloning experiments. They are produced by bacteria, and they help the bacteria to protect the bacteria from bacteriophages. Or these are restriction enzymes. They are also molecular scissors. So yes, so the correct answer for this question is option C. So RNA polymerase is the main enzyme which is used for transcription. So as it was discussed in your lectures that RNA polymerase. In prokaryotes, there is a single RNA polymerase, and it is responsible for synthesis of all type of RNA. The eubacterial RNA polymerase is named as holoenzyme. Why is it called holoenzyme? So it is made up of 5 subunits, and this alpha dash beta, two alpha subunits, and one beta and beta dash subunit. When it joins with a sigma factor, the entire complex is called as holoenzyme. So holoenzyme is core enzyme plus the sigma factor. It contains 5 subunits. The alpha is assembly of core enzyme, beta dash performs all enzymatic and catalytic function, and sigma helps in the recognition of the promoter sequence. So we will move to the next question. The DNA sequence is read by an RNA polymerase that produces a complementary anti-parallel RNA strand. So the complementary anti-parallel RNA strand that is synthesized by RNA polymerase during transcription is known as. So what is the name for this anti-parallel RNA strand? Hexatranscript, secondary transcript, primary transcript, tertiary transcript. So what will happen during transcription? A small region of the DNA will open up, right? It will open, and then RNA polymerase will act on one of the strands, or it will act on the template strand, and a complementary RNA strand will be synthesized. So this RNA strand which is synthesized, this RNA strand which is synthesized, or this is called the primary transcript, right? So this is called the primary transcript. So you will often encounter this term in books, in textbooks, that it is written as the RNA transcript that is synthesized. Or so the primary so that is why the antiparallel RNA strand which is synthesized, it is called the primary transcript. Okay. This does not require much explanation, so is option C. Okay. Now I'm moving to the next question. So the next question, transcription factors gets bound and phosphorylates polymerase II to initiate transcription in the presence of ATP, and it also functions as helicase that unbinds DNA. So the transcription initiation, the transcription initiation in eukaryotes is different from that in prokaryotes. So for the transcription initiation in eukaryotes, there are different transcription factors that are required for the RNA polymerase to be recruited on the DNA, on the transcription unit of the DNA. So there are several transcription factors. So starting from transcription factor 2D, then it will help in the recruitment of transcription factor 2A, then B, then G, F, H. So like this, there are multiple transcription factors that are involved. So this is actually a fact-based question. So we have to understand the initiation of transcription in eukaryotes. So how does transcription initiation take place in eukaryotes? So in prokaryotes, the sigma factor is responsible for recognition of the promoter region, so that the RNA polymerase can recognize the promoter region of the DNA. For that, the sigma factor is required, and once the sigma factor binds to this sigma factor recognizes the promoter region, and the RNA polymerase starts synthesizing the new RNA transcript. After 10 to 15 nucleotides, ribonucleotides that are synthesized, this sigma factor is released. The sigma factor is released, and the rest of the function can be carried out by the other subunits of the RNA polymerase, while in case of eukaryotes, there are several transcription factors that are required for the initiation of transcription. So this was discussed. So yeah, the answer is transcription factor 2H. We will go through this transcription initiation in eukaryotes. So this is directly taken from their lecture slides. So here you can see that the transcription, basically the promoter region. So the promoter region is a DNA sequence. You know the promoter region, right? The promoter region is it is a region of DNA, or it is a DNA sequence. It is a DNA sequence which is recognized by the RNA polymerase, and it is this region where the RNA polymerase will bind and start the transcription process. Okay, so this promoter region has certain consensus sequences. So in case of the prokaryotes, the consensus sequences were the Tata box and Prigno box. Here in case of eukaryote, there is Tata box and then there is INR. This consensus sequences. So at these regions, the RNA polymerase will bind, and then these transcription factors are sequentially they bind to the Tata box DNA to form a pre-initiation complex. So what is the pre-initiation complex? So the RNA polymerase, along with all these transcription factors, together the transcription factor and RNA polymerase, they form a pre-initiation complex on the promoter region of the DNA. So first, what happens? So this has been shown in this figure. So first, there will be TF2D, then followed by TF2A, TF2B, TF2F, TF2E, and TF2H. Okay, so at last, when TF2H binds, it phosphorylates polymerase 2. It phosphorylates polymerase 2 to initiate transcription in the presence of ATP. So the TF2H is the transcription factor which will phosphorylate. So that was asked in the question. So which is the transcription factor which phosphorylates polymerase 2 and initiate transcription? So it is transcription factor 2H. So transcription factor 2H is the last transcription factor to bind to the RNA polymerase, and this transcription factor will phosphorylate the C the RNA polymerase has the C terminal tail, and this once this phosphorylation takes place, it acts as a signal that now the RNA polymerase can move on to the elongation phase and synthesize a new RNA strand. Okay. So here, there was a table that was there that was discussed in the lecture. So you can go through this table. So it describes it describes the function of all the transcription factors that are involved in the transcription initiation of eukaryotes. So transcription factor 2D. So this transcription factor 2D, what is its function? So it is the first transcription factor to bind to the RNA polymerase, and its function is to recognize the promoter. So it will recognize the Tata box, and then there are TAFs. So TAFs are the transcription binding protein associated factors. So these factors will bind with the TBP, and it will help the RNA polymerase to recognize the core promoter. Okay, then this is followed by recruitment of transcription factor 2A. So what does transcription factor 2A do? So this transcription factor 2A, it stabilizes the binding of TBP and the transcription associated factors to the RNA polymerase, right? And then the transcription factor 2B is recruited. So it helps in the RNA polymerase 2 and transcription factor 2F recruitment, and also help in the start site selection. So this transcription factor 2B, what will it do? It will help in the recruitment of RNA polymerase 2 and also the next transcription factor which is required, which is transcription factor 2F. And this transcription factor 2F is the transcription factor which helps the RNA polymerase to bind to the promoter. So you have the RNA polymerase. So the RNA polymerase. So this is the RNA polymerase. It is the claw-like structure. So this RNA polymerase will first bind to transcription factor 2D, then followed by A, then followed by B, and then followed by transcription factor F, transcription factor 2F. So once this transcription factor 2F binds to this complex RNA polymerase along with all these other transcription factor, it will help the RNA polymerase to recognize the start site. So the start site of the of transcription. Start site of the transcription is that site which where the RNA polymerase will initiate the transcription process. Okay, so this is the transcription factor 2F. It will help the RNA polymerase to bind to the promoter. And then transcription factor 2E helps in TF2H recruitment, modulation of TF2H, helicase, ATPase, kinase activities, all the helicase and ATPase and kinase activities are done by this transcription factor 2E. And then last transcription factor is the transcription factor 2H, which helps in the promoter melting with helicase activities, promoter clearance by phosphorylation. So what is promoter clearance? So this process is this term, what it describes is so promoter clearance means when the RNA polymerase will move from the ahead of the, or it will move from the promoter region and enter the coding region of the transcription unit. So that is promoter clearance. Okay. So remember that this transcription factor 2H has helicase activity, and it also, what is the other function of transcription factor 2H? It phosphorylates the RNA polymerase. So it phosphorylates the RNA polymerase and helps the RNA polymerase to move from the promoter to the coding region of the transcription unit. So that is that process is called as promoter clearance. Now coming to the mRNA processing. Okay, so what happens? So just I was discussing during in the beginning of the today's lecture that the mRNA which is synthesized in the eukaryotes, it requires processing. Requires processing, and why? Because the mRNA has to be transported from the nucleus to the cytosol, and since this RNA is a single-stranded structure, it may get degraded by the various ribonucleases, exonucleases, endonucleases, which are present in the cytosol. So there has to be certain modifications taking place, and also this it also provides stability to the mRNA and identification of the mRNA by the ribosomal subunits, which is present in the cytosol for subsequent translation of the mRNA into protein. So there are what is the mRNA processing? There are certain post-translational modifications which take place in the mRNA. So there are three main processes which take place, which is capping, splicing, and tailing. So you must have read about this in basic biology course in class 12 also. So what happens is this mRNA which is synthesized, it will have a 5' end and a 3' end. This 5' at the 5' end, what happens? A 7-methyl guanosine, 7-methyl guanosine triphosphate molecule is added. So this is it adds a cap at the 5' end of the mRNA. Then there is a poly A tail which is added to the 3' end of the mRNA, and the eukaryotes. One more difference between the eukaryotes and prokaryotes is that the mRNA that is synthesized in eukaryotes, they have non-coding regions between the coding regions. So there are introns. So introns are the regions which do not code for any protein, and there are intervening regions which are present between two exons. So these introns are removed, and these are the introns. These introns are removed and by a process known as splicing, and the subsequent exons are joined to each other. So that then we get a completely processed mRNA which is ready for translation. Okay. So what is the order in which this the modifications in mRNA take place? So remember that the order is this one. So first capping takes place, then splicing, and once the 3' end of the 3' end of the mRNA is present, the tailing, this 3' end is acted upon by other enzymes, which adds polyadenylate residues to the three prime end of the mRNA. Okay, so the answer is option A, 1, 2, 3. Okay. Okay, so the first RNA processing event is capping. So what happens in capping? So in capping, there is addition of modified guanine base to the 5 prime end of the RNA, joined to the RNA transcript by an unusual 5 prime linkage. So there are this capping process that has multiple steps and is acted upon by several enzymes. So first there is this for phosphorylase enzyme will come, and then there will be guanylyl transferase enzyme, which will add a methyl group to the guanine residue, and ultimately, after this modification, there is another guanylyl transferase will act, and then there is methyl transferase enzyme. So this methyl transferase enzyme will transfer a methyl group to the guanine, and this modified guanine residue which is produced is added onto the 5 prime end of the mRNA. Then what happens? So once the RNA is capped, so the RNA is capped as soon as it emerges from the RNA exit channel of the polymerase. So if you look at the structure of the polymerase, there are several channels in the RNA polymerase. So there is an RNA exit channel. So from here, what happens? This RNA exit channel, the RNA that is being newly synthesized is comes out of the RNA polymerase. So after capping, what happens? There is dephosphorylation of the serine pipe. So there is dephosphorylation of the serine pipe within the tail repeats, and this dephosphorylation acts as a signal for the dissociation of the capping machinery. So as I was discussing that the capping machinery, there are several enzymes which helps in the addition of this modified guanine onto the 5 prime end, and so when this serine fiber is phosphorylated, it acts as a signal for the dissociation of this capping machinery and further phosphorylation or further phosphorylation. So the next phosphorylation that takes place is at the serine 2 residue. So at the serine 2 residue, when phosphorylation takes place, it causes the recruitment of the machinery needed for RNA splicing. So the RNA splicing is again a series of different reactions that takes place. There is spliceosome complex, and there are several snRNAs and other molecules which are involved in this splicing process. And at the end, what happens? The final RNA processing event is polyadenylation. Polyadenylation of the 3 prime end of the mRNA. Okay. So remember that the order is 1 followed by 2 and 3. So tailing is the last mRNA processing step. Okay. So I hope things are clear till this point. Okay. So yes, this is what I was explaining here. So this RNA polymerase has a C-terminal tail, and this C-terminal tail is composed of a heptapeptide. Heptapeptide means there are seven residues which constitute the C-terminal tail of the RNA polymerase. So these are YSPT, SPS. So the phosphorylation, as we know that phosphorylation, we had also learned in earlier lectures that phosphorylation takes place in serine, threonine, right? Threonine residues and the serine residues. So first, what happens is serine, threonine, and tyrosine residues. Okay, so when the serine 5 residue is phosphorylated, this it acts as a signal for. So this is the pre-initiation. So in the pre-initiation, as a step, or the none of these C terminal, none of these residues in the C terminal chain is phosphorylated, right? And then when the serine 5 is phosphorylated, this phosphorylation is what is done by this transcription factor 2H. Okay, so when this serine residue is phosphorylated, it acts as a signal of promoter escape or promoter clearance. Okay, and following the promoter escape, this signal is also or this phosphorylation at the serine 5 residue. It is also a signal for the recruitment of the capping machinery. Okay, so the various enzymes for the capping process is recruited following this phosphorylation at the serine 5 residues. And then when in the next step, when this serine 2, when this serine 2 is phosphorylated, this serine 2, once it is phosphorylated, the capping machinery will move off from this heptapeptide, and then it will act as a signal for elongation. And and when the serine 2 is phosphorylated, the RNA processing factors that are recruited, there's various factors that are involved in the splicing or the removal of this introns from the mRNA is recruited once this serine 2 is phosphorylated. So this figure is from the book J.T. Watson, Molecular Biology of the Gene. So here you can understand how this C-terminal domain of this RNA polymerase is involved in the transcription process. Okay. So we will move to the next question. So in eukaryotic organisms, 45S precursor is formed by RNA polymerase 1 and processed in 90S 3-ribosomal nucleolar complex to give. So thus, now I was discussing that the RNA polymerase 1, it transcribes, RNA polymerase 1 transcribes rRNA. Okay, so now in rRNA also there are several rRNAs which you have seen when we were discussing ribosome, the structure of ribosome. So there are in eukaryotes there are three main rRNAs, right? These are 18S rRNA, 28S rRNA, 5.8S rRNA, and the 5S rRNA. So there are rRNAs which constitute the larger subunit of the ribosome, and rRNAs which constitute the smaller subunit of the ribosome. So in this question, what is asked is that the 45S precursor. So the eukaryotic ribosome is 80S. It is a 80S ribosome, and it has a 45S and a. So here the 45S precursor. What are the rRNAs? How this 45S precursor, gives rise to, or the 45S precursor that is produced, how is it gives rise to, or which what are the rRNAs which are arising from the 45S precursor that has been on. So this rRNA processing in, so yes, the answer is actually option number D. So so if you see this figure, this is again from I've directly taken it from the lecture slide. So this is the 45S rRNA. So this is the precursor rRNA, 45S. And this 45S rRNA, processing of this 45S rRNA gives rise to three main rRNAs, which are 18S rRNA, 5.8S rRNA, and 28S rRNA. Okay, so in eukaryotes, the nucleolus is the center of processing ribosomal RNA. And also remember that the RNA polymerase 1 is actually present in the nucleolus. Uh, so, so the uh, So the center of processing of ribosomal RNA is the nucleolus, and the 45S precursor is formed by RNA polymerase 1. So the precursor that is formed is 45S rRNA, and then this is processed. This is processed in the 90S autosomal nucleolar complex to give rise to 18S, 28S, and 5.8S rRNA. So there is tight coupling of RNA processing ribosomal activity. But remember that this 5S rRNA, 5S rRNA is not formed by the processing of the 45S precursor RNA. This 5S rRNA is transcribed by RNA polymerase 3 from a separate gene. And this this 5S rRNA is transcribed by RNA polymerase 3. So RNA polymerase 3 transcribes for tRNA as well as 5S rRNA. Okay, the precursor 5S rRNA, so it is the only rRNA which is not synthesized by RNA polymerase 1 but by RNA polymerase 3. So the precursor RNA undergoes methylation at more than 100 bases from 1400 nucleotides at 2' hydroxyl group. So there are several modifications which are present in the precursor RNA, and there are modified bases which are present in the precursor RNA, such as uridine, pseudouridine, and followed by a series of cleavage reactions and modifications. We get a structure called SNU RNAs, or small nucleolar RNAs. And in each, the entire processing involves pre-rRNA, 170 non-ribosomal protein, 70 SNU RNAs, and are supposed to be remnants of lysosomes. So remember that from 45S rRNA, there is this processing of this 45S rRNA in the 90S ribosomal, pre-ribosomal nucleolar complex. There are 3 rRNAs that are produced, which are 18S, 5.8S, and 28S. Okay. Okay, so we will move to the next question. Okay. So the next question is basically, which of the following statements regarding splicing in eukaryotes is correct? So what happens in splicing? So in splicing, the introns are removed and the exons are joined. The alternate exons are joined to each other. So there is the spliceosome complex, which helps in the splicing process. So the option is what? There are several reactions in the splicing process involving hydrolysis of ATP. Exons are spliced out and introns are retained in the mature mRNA transcript. So this is straight away you can eliminate this option, because by the very definition of splicing, you know that the exons in splicing, what happens is the exons are not spliced out. They are the introns. Introns are spliced out, and the exons are retained in the mature mRNA transcript. So this option is incorrect. Splicing takes place in the cytosol. This is also wrong. So just now we learned the concept that the entire mRNA processing takes place in the nucleus before the RNA can be transported out of the nucleus into the cytosol. There has to be several modifications taking place. So this option where it says that splicing takes place in the cytosol is incorrect, right? Then small nuclear RNAs are retained in the mature mRNA transcript. So this is also wrong. I believe so mature mRNA transcript will only have the exons. It will neither have introns, it will other small nuclear RNAs. Okay. So yes, the correct answer is option A. So there are several small nuclear RNAs which are involved in this process, U6, U5. There are a cascade of reactions that take place one by one for splicing of the introns. So option number A is the correct option. So here you can go through this slide. So what happens? These introns are non-coding nucleotide sequence within a gene that do not code for protein and don't appear in the final mRNA molecule. and these introns are therefore removed by splicing. Okay. The protein coding sequence of gene is known as exons, and these exons are interrupted by introns. So the vast majority of eukaryotic genes are interrupted with non-coding regions. So the exons are interrupted with introns, and these introns have to be spliced out. However, histone protein coding genes in vertebrate is among few exceptions. The occurrence of introns varies in eukaryotic species. So there are always exceptions, right? So each species will lack introns, and many genes in eukaryote carry a dozen of them. Then there are a few bacterial and archaeal genes which are exceptions in the sense that they have introns, and introns can vary in length from 50 to 20,000 nucleotides. In higher animals, as humans, introns are more than exons. So there are four different classes of introns: group one, group two, group three, and group four. So group one and so how do these different classes of introns differ in the splicing machinery? Were discussed in your lectures. You can go through the lecture once again. So basically, the group one and group two introns are self-splicing introns and does not involve any protein enzymes. And the group three introns are called the spliceosome introns that are not self-splicing, and there are introns that require ATP for splicing. So based on how the splicing machinery, what is the function, how does the splicing machinery function? They are grouped into four different classes. Okay, now we will come to the concept of operon. Concept of operon in prokaryotes. So, so one difference between the prokaryotes and eukaryotes is that the prokaryotic mRNA is polycistronic, while the eukaryotic mRNA is monocistronic. Okay, it is monocistronic. So what do you understand by the term polycistronic and monocistronic? So polycistronic are so prokaryotes have polycistronic genome or polycistronic mRNA, and eukaryotes are monocistronic. So polycistronic basically means that more than one polypeptide will be synthesized from a single mRNA. Okay, so you have a single mRNA, and from the single mRNA, you can have more than one polypeptide. Okay, so there can be several polypeptides that are synthesized. For example, there are three different polypeptides that are getting synthesized from a single mRNA. And then monocistronic. So we will discuss this. We will understand this when we discuss the concept of lac operon in bacteria. And monocistronic means that from a single mRNA to single mRNA, only a single polypeptide is synthesized. So yes, single polypeptide is synthesized. That is monocistronic. And also another thing is, uh, so what is an operon now? Okay. So operon means so when there are multiple genes which are controlled by a single promoter and a terminator, okay, which are under the control of a single promoter. So a single promoter is controlling the expression of more than one gene. That is called the operon. And why is it? So this operon is mainly present in prokaryotes. And why is it required? This is required because all these genes are involved in the same. They can be involved in the same biochemical pathway, or all the three proteins are involved in are related to each other in performing a common function. Okay. or involved in a particular biological process. So that is why these three genes are together. They are controlled by a single promoter region of the DNA. So, so in this question, the so you know how an operon looks, right? So the operon will have a promoter region. It will have a so first there will be a regulator gene. Then there will be a promoter region, then operator region, then there will be structural genes, and then there will be a terminator region. Okay. So X is a nucleotide sequence that acts as the site for RNA polymerase attachment and transcribe. So X is a nucleotide sequence that acts as a site for RNA polymerase attachment and transcribes a gene or a group of genes. Okay. So a group of genes. The Y segment. Y is a segment of DNA that acts as a site for repressor attachment. So you have to identify the X and Y. So the X is a nucleotide sequence. So it is a sequence of DNA where the promoter where the it is a sequence of DNA where the RNA polymerase will bind. Binds, and Y is a DNA sequence where what is happening where it is a site for attachment of repressor protein. So what is this region called? So this DNA region where RNA polymerase binds is called the this is called the promoter region. So it is a region of the DNA sequence which is recognized by RNA polymerase uh, and is the site where transcription initiation will take place, right? And repressor protein, what it does, repressor protein, what is that region of the of an operon, or what is that region of a DNA where the repressor protein binds? So that region of the DNA is called the operator region. So operator region is a DNA sequence which is recognized by the repressor protein and this and this region it lies ahead of the promoter region, and it is this region where the repressor protein binds and prevents RNA polymerase from transcribing the that part of the gene. If you look at the structure of an operon, so there is a promoter region. So first there is a regulator gene. Basically, this regulator gene, what it does, it codes for a repressor protein, or it can either code for a repressor protein or an activator protein. So it will code for a protein which will control the transcription of a group of genes, which will control the transcription of a group of genes. So it can either be a repressor protein, it can be an activator protein, and based on that, there are different. That this operon is classified as positive operon or negative operon. Okay. Then this regulator gene is followed by a promoter region. So this promoter region is the region where the RNA polymerase will bind. So this is the promoter region. Then this promoter region is followed by an operator region. So first is the operator region actually, and then followed by the promoter region. First operator region, followed by promoter region, and then there are structural genes. I'm sorry, first there will be the promoter region, then this is followed by the operator region, and then the structural genes, and then there will be the terminator. So what happens if the regulated gene is, for example, it is coding for a repressor protein, this repressor protein will bind to the operator region. It will bind to the operator region and it will prevent the RNA polymerase from transcribing the structural genes. Okay, so this repressor protein, so if it is a repressor protein, it will inhibit the RNA polymerase from moving ahead. But if it is an activator protein, it will enhance the binding of this RNA polymerase to the promoter and transcribe the structural genes. Okay, so the site for this answer is actually X is the promoter and Y is an operator. Okay. So these were discussed in the lectures as well. So we will go through this lecture slide where this concept has been discovered, where this concept has been described. Okay, so the basic structure of an operon is that it has a promoter, it has an operator and structural gene. Promoter, operator, and structural genes. A regulatory gene is not part of the promoter, is not part of an operon. Regulator gene is not part of an operon, but it is involved in controlling the transcription of genes which are a part of an operon. Okay, so you can see here, this is a general structure of an operon which has been depicted in this figure. So there is a regulatory gene. This regulatory gene codes for a regulatory protein, and this regulatory protein can be an activator or it can be a repressor. And then this regulatory gene is followed by a promoter, and then promoter followed by an operator, and then there are structural genes. Okay, so at this operator, why is it called operator? Because at this site, it is at this DNA sequence where the repressor or the activator protein will bind. So this operator is the region where this repressor protein binds and it will prevent the RNA polymerase from moving ahead. And then there are structural genes. So there can be 2, 3, 5, 6 different or more than one different structural genes. So there can be a group of genes which are present in the structural genes. So the regulatory gene codes for protein called regulatory protein, which can either act as repressor or activator, which controls the operon. But it is not part of an operon because it has its own promoter. So the regulatory gene has its own promoter. So remember that regulator gene is not part of an operon. Why? Because it has its own promoter. And by the very definition of operon, we know that all the genes which are controlled by a single promoter will constitute an operon because this regulated gene has its own promoter. So this regulated gene is not part of an operon. Okay. So promoter is a nucleotide sequence that acts as a site for RNA polymerase attachment and transcribes the gene. So it is a DNA sequence which acts as a site for RNA polymerase binding. And following the binding of the RNA polymerase, it will transcribe the gene. And this promoter is the RNA polymerase recognize the promoter and then initiate transcription. So operator is the segment of DNA which is present between the promoter. So operator is present between the promoter and the structural genes. So operator is present between the promoter and structural genes. It is that segment of DNA which is acted on by or which is the site of repressor attachment. So it is the site of repressor attachment and it is generally situated in between the promoter and the structural gene of the operon. It is generally situated. It can be overlap with the promoter sequence, but it is usually what is present in the promoter and structural genes. And the structural genes at the end of the operon, there are set of structural genes. And these genes are transcribed into a single mRNA and finally translated into proteins. So the mRNA will be there will be a single mRNA which will be synthesized by these structural genes. Will code for a single mRNA, and then this mRNA from this mRNA there will be different polypeptides that will be synthesized from the single mRNA. They are transcribed into proteins. So these mRNA will have start and stop codons. So we will learn about the start and stop codons when we discuss about translation. So between these different structural genes, there are start and stop codons. So when the ribosome binds to this mRNA and starts translating it into protein, there will be different polypeptides that will be synthesized. Okay. Now, how does lactose promote transcription of LAC-Z? So, basically, if you have gone through the lectures, I hope you have understood the basic concepts of lac operon. So the lac operon or the lactose operon is present in the E. coli bacteria. And what happens here is this lac operon is involved in breaking down the lactose, which is present in the environment into glucose and galactose. So this glucose can then be utilized as an energy source. So if the bacteria is kept in a medium, or it is kept in an environment which lacks glucose but has lactose, then rather than utilizing glucose as a carbon source, now it will start utilizing lactose. Now lactose is a disaccharide, so this disaccharide has to be broken into monosaccharide, which can then be used as an energy source. So there are several enzymes which are involved in this process, and all these different enzymes that are involved in this process constitute they are present in the bacteria in the form of the lac operon. Okay, so what happens is this lac operon, it is an example of negative inducible operon, right? Negative inducible operon. Why? So it is negative because here the regulator gene codes for a protein which is a repressor. Regulator gene codes for a protein which is a repressor. Why is it a repressor? Because it prevents the RNA polymerase from transcribing the structural genes, and it is inducible. Why it is inducible? Because normally this lac operon will remain off. Normally it will remain off in normal conditions, or when there is glucose in the media, this lac operon will remain off, while it will be turned on when lactose is present in the media. So it is an inducible operon. Inducible means it has to be induced by something, or it has to be turned on by some factor or by a signaling molecule. So that is why it is inducible. It normally remains off. It has to be switched on. So it will so this machinery is present for it helps the bacteria to conserve its energy, because when the media in which the bacteria is being cultured, if this media has glucose as the energy, media has glucose as the energy source, then there is no need for this lac operon to function. But this lac operon will only be required if the glucose is absent in the media while lactose is present. Okay. So 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 how does lactose promote transcription of LAC-Z? So basically what happens in lac operon, this lactose is converted into allolactose, and this allolactose is an inducer for the lac operon. Okay, so it is an inducer for the lac operon, which means that this allolactose, it is the signaling molecule which binds to the repressor protein. So if you look at the structure of an operon, so there is a regulatory LAC-I gene. Okay. There is a LAC-I gene, then there is a promoter, then operator, and there are three structural genes: LAC-Z, LAC-I, and LAC-A. So these LAC-Z codes for different enzymes: beta galactosidase, then LAC-Y codes for permease, LAC-A codes for transacetylase. So these codes for different there's a single mRNA that is produced, and then from this mRNA, there are three different polypeptides which are produced. So what happens? This LAC codes for a repressor protein. So this is an active repressor. So this because this is an active repressor, it goes and binds to the operator region. It will go and bind to the operator region, and it will prevent the RNA polymerase from, or it will prevent the RNA polymerase from transcribing the structural genes. Okay, it will not allow. It will prevent the RNA polymerase from moving ahead. Now when So now when allolactose is present in the media, what allolactose does, it binds to the repressor. It binds to the repressor. So this is the allolactose, and for example, this allolactose binds to the repressor, and it causes a conformational change in the repressor protein. So once there is a conformational change in the repressor protein, the site of the repressor which was binding to the DNA that gets altered, and as a result, it can no longer bind to the DNA. It can no longer bind to the DNA. Now the DNA becomes, once it is this operator region is free, this RNA polymerase can move ahead and transcribe the structural genes. Okay. So here allolactose is acting as an inducer. So allolactose is the inducer, or it, it is a signaling molecule which is causing the lac operon to which is switching on the lac operon. Okay. So what does lactose do? So option A says that the lactose binds directly to the polymerase and increases efficiency. So this is wrong. So lactose is the inducer. The lactose binds, this inducer binds to the repressor protein. It does not binds to the polymerase. Okay, so option A is incorrect. Lactose binds to a repressor protein and alters its conformation to prevent it from binding to the operator and interfering with the binding of RNA polymerase. So this option is absolutely correct. So this lactose, what it does, it binds to the repressor protein and causes a conformation change in the repressor protein, so that it can now no longer bind to the operator region. Okay, so option B is the correct option. Now lactose is an activator protein, is not an activator protein, it's an inducer. The option C is incorrect, which can help the RNA polymerase to bind to the promoter and begin transcription. This is completely incorrect. And then lactose prevents premature termination of transcription by directly binding and bending to the DNA. So this is also incorrect. Okay. So the answer is option B. So you have taken from this lecture slide. So this lac repressor. So the lac repressor strongly. So when lactose is not available. So but as I was explaining, that when lactose is not present in the media, the operator, this lac repressor, it will strongly bind to the operator and stop RNA polymerase from initiating transcription. However, the lac repressor loses its capacity to bind DNA when lactose is present. Okay, so when lactose is present, this repressor will lose its capability to bind to the operator and floats away, making it possible for RNA polymerase to transcribe the operon. When lactose or allolactose is present, this way it will bind to the repressor. It will change its shape and it will prevent it from binding to the operator, so that the RNA polymerase can easily bind with the promoter and cause the transcription of subsequent genes. So allolactose acts as an inducer. Okay, so this has been explained in this figure as well. So you can see here in this figure that this is the repressor. This repressor has been shown as in this brown colored structure, protein cartoon. So this repressor is binding to the operator. It is preventing the RNA polymerase from moving ahead when lactose is not available. While when lactose is present, that there is a change in the conformation. So first the conformation was like this. When shallow lactose is binding, its conformation has changed. Now this conformation does not allow the repressor protein to bind to the operator, and now the operator is free, and RNA polymerase can move ahead and transcribe the genes. Okay. Now, what molecule acts as a secondary messenger to the lac-operon regulatory system at low glucose levels? So, what happens is that low glucose levels, so there is one positive regulation, and this positive regulation is done by the cap protein. Okay, so there is this cyclic AMP. So this cyclic AMP, remember, is the secondary messenger which which acts at low glucose levels. So the levels of glucose and the levels of cyclic AMP are inversely related. If the glucose levels are low, the cyclic AMP levels will be high. When the glucose levels are high, the cyclic AMP levels will be low. So yes, this is option A is correct option, and we will look at this slide. It was discussed in the lectures as well, so we will understand this slide once again. So you can see here that ahead of the promoter, you have to understand that other than this promoter, operator, and the structural genes, just upstream of the promoter, there is another site. There is another site which is called the cap binding site. Now, cap is a protein which is called catabolite activator protein. Catabolite activator protein. So this protein binds at a site which is upstream of the promoter, which is called the cap site. Now this cap protein can only bind to this cap site when this catabolite activator protein is bound to cyclic AMP. So only when CAP, this catabolite activator protein, only when this cap protein is bound to the cyclic AMP, only then this complex can bind to the cap and promote the or enhance the activity of the RNA polymerase. So this is a positive regulation of the lac operon. So this cyclic AMP levels determine whether this cap protein can bind to the cap site. So when the glucose level, so when the glucose levels are high, so if the glucose levels are high, you do not need this lac operon to remain active, right? You would not want this operon to be turned on in presence of high glucose level, and this operon system would be essentially off. The operon system. So how is it done? So this is done by this cyclic AMP molecule. So when glucose levels are high, the cyclic AMP levels will be low. And now since our cyclic AMP levels are low, it will not be able to bind to the CAP protein. And now, since it does not bind to the CAP protein, this CAP protein cannot bind to the cap binding site on its own. So it cannot bind to the cap binding site on its own. It can only bind to the cap binding site when it is in complex with the cyclic AMP. So as a result, what will happen if my cyclic AMP levels will be low and my lac operon will remain turned off. My lac operon will remain turned off in absence of the cyclic AMP, in absence of cyclic AMP. While when my glucose levels are low, the cyclic AMP levels will be high. Now this cyclic AMP will go and bind to the cap binding site, and it will enhance the RNA polymerase, or it will activate the RNA polymerase, and my lac operon will remain turned on. So that is what has been explained, shown through in this figure. So glucose absent, lactose absent, no transcription. Glucose absent, lactose present, high transcription. So what will happen when the glucose is absent and lactose is present? The transcription will be very high. Why? The transcription will be very high because when glucose is absent, so when lactose is present, so what will happen when lactose will be present? So lactose will first of all it will bind to the repressor, and it will change its conformation so that it can no longer bind to the operator, and the and the RNA polymerase can transcribe the structural genes. Okay, and at the same time, what will happen? My cyclic AMP levels will also be very high. So both the things are acting together. This my cyclic AMP levels are high. As a result, what is happening? The catabolite activator protein can bind to the cap site and it so this cap protein basically acts as an enhancer. It enhances the activity of the RNA polymerase, and so the lac operon will start acting very or it will be or the transcription rate of this lac operon will enhance by several folds. And at the same time, what is happening? My repressor protein is also getting inactive. Repressor protein has become inactive. Why? Due to the binding of this lactose. Binding of the lactose, my repressor protein has become inactive. So my operator region is also P and the RNA polymerase can move ahead. But what will happen when glucose is absent? Lactose is absent. So glucose and lactose both are absent. There will be no transcription of the lac operon. Right? If my lactose is also absent and glucose is also absent, there will be no transcription. So glucose is absent, the cyclic AMP levels will be high. But the cyclic, but the operator region will be occupied by repressor. So the RNA polymerase cannot move ahead and transcribe the downstream genes which are present downstream to it. Okay, that is what has been explained in this figure. So the cyclic AMP levels are high because the glucose levels are low. So cap is active and will be bound to DNA. So cap will go and bind to the DNA. However, the lac repressor will also be bound to the operator because there is no inducer, and it will act as a road blocker that will prevent RNA polymerase from transcription. Okay. Now, coming to the next question, the bacteria utilize glucose preferentially over other sugars through a mechanism called. So this mechanism, what is this mechanism called? The options are operon repression, enzyme repression, catabolite repression, and catabolite induction. So the answer is catabolite repression. So what happens if the bacteria is grown in a media which has both glucose and other sugar substrate? For example, it has glucose, lactose. Then the bacteria, what will it do? It will preferentially utilize glucose and not, and it will once all the glucose has been utilized, then it will transition to utilize lactose from the media. So, and how is it done? So this mechanism is called catabolite repression. So what is this mechanism? Let's understand this. So this E. coli and many other bacteria metabolize glucose preferentially in the presence of lactose and other sugars. So even when other sugars are present in the media, still it will preferentially utilize glucose. And so when glucose is available, this process is called catabolite repression. Okay. So when glucose is available, the genes that participate in the metabolism of other sugars. Just give me a second. Okay, so let's start the discussion. Okay, so we were discussing about catabolite repression. So in catabolite repression, so what do you understand by catabolite repression? So what happens is when glucose is available, the genes that participate in metabolism of other sugars are repressed. This phenomenon is known as catabolite repression, and this catabolite repression results from positive control in response to glucose. Okay, so the positive control is accomplished. So in response to glucose, there is a positive control on the black operon. Okay. So this positive control is accomplished through the binding of a dimeric protein called the catabolite activator protein, which binds to a site which is around 22 nucleotides long, and it is located slightly upstream of the promoter of the lag gene. So there is the promoter region, and this promoter region is upstream of the lag gene. So it is the promoter, the cap binding site is upstream of the promoter region, and it is at this cap binding site where the catabolite activator protein will bind. And before cap bind to DNA, it must form a complex with cyclic AMP. So this is what I was explaining. So only when the cyclic activator protein is in complex with the cyclic AMP, only then it can bind to the cap binding site and enhance the activity of the RNA polymerase. The concentration of cyclic AMP is inversely proportional to the level of available glucose. Okay, so let's understand this. This is directly taken from the lecture slides which were discussed. The lag promoter is a weak promoter. Okay. So you have to understand this that lag promoter is a weak promoter, and it does not uh, uh, does not bind to RNA polymerase more efficiently on its own. So on its own, it has a very weak affinity to the in binding to the promoter region. It won't be able to accomplish much more without the help of the catabolite activator protein. So when the catabolite activator protein will bind to this promoter region, the transcription levels will be enhanced before the lagoperon promoter. So the E. coli produces cyclic AMP as a hunger signal in low glucose condition. So in low glucose conditions, you need to turn on the black operon. Okay, so that the signaling molecule here is the cyclic AMP, which is acting as a hunger signal, and this cyclic AMP levels will increase. As a result, it will bind to the cap and stimulate transcription. So cap is inactive without cyclic AMP. Only when glucose levels are low, the cyclic AMP levels are high, and as a result, this catabolite activated protein now it will become active and it can bind to the cap binding site of the DNA. So therefore, high amounts of lac operon transcription are only possible without glucose. So without glucose only, there will be high amounts of lac operon transcription. Okay. This method ensures that bacteria only activate the lac operon and begin using lactose after exhausting their primary energy source. So this is called catabolite depression. So you can see over here in this figure, low glucose levels means there will be high cyclic AMP levels, and once there is high cyclic AMP, it will go and bind to the catabolite activator protein. Then this catabolite activator protein now becomes active and can bind to the cap binding site and enhance the activity of the RNA polymerase, resulting in high levels of transcription. Now, when the cap is not bound to the cyclic AMP, it is inactive. It cannot bind to the catabolite activator from this cap binding site, and as a result the activity of there will be low transcription. Okay, so when glucose levels are high, production of cap efficiently reduced, and cyclic MP now cannot attach to the cap. As a result, the cap remains inactive and unable to bind to the DNA. RNA polymerase will bind to the to the promoter, but as it does not get extra help from the catabolite activated protein, the levels of transcription will be low. Now coming to the next question, in a lac operon, a mutation is created so that the lactose cannot bind to the repressor. So if the lactose cannot bind to the repressor, then what will happen? This is a very easy question. So if the lactose is not, if my lactose is not binding to the repressor, then the repressor will not become inactive. The repressor will always remain active. So if my repressor is active, then even in the presence of lactose, the repressor will still go and bind to the operator side, and it will prevent transcription of the lac operon genes, right? So if my lactose, if my lactose is not able to bind to the repressor. By repressor, so the lactose is present in the media, but still the lactose cannot bind to the repressor protein. So in that case, my repressor will remain active. My repressor will remain active. It will go and bind to the operator region. So this is the operator region. It will go and bind to the operator region, and it will prevent the RNA polymerase from transcribing the genes. Okay, so as a result, what will happen, even if lactose is externally provided in the media, the beta galactosidase will be overexpressed. This is wrong. Why will it be overexpressed? Because my repressor protein is blocking the path of the RNA polymerase. So how will the beta galactosidase be overexpressed? So this is wrong. Lac-I repressor will remain inactivated. Lac-I repressor will remain inactivated. This is wrong. Lac-I repressor will be inactivated by lactose protein, but the lactose protein by the lactose signaling molecule, but the lactose molecule is not able to bind to the lac repressor. So this lac-I repressor will always remain active. Okay, the expression of lac operon will remain unaffected. This is also wrong. The only correct answer is the lac-I repressor will continuously prevent expression. It will lac-I repressor, it will prevent expression, because it will not allow the RNA polymerase to move ahead and transcribe the structural genes. Okay. Answer is option D. Now select the most appropriate option about the regulatory mechanisms listed below for the regulation of the TRP operon. So the TRP operon is another operon system which is present in the E. coli. So the correct answer is option A, actually, repression and attenuation. And why, what do you mean by repression and attenuation? We will just see in the next few slides. So there are two mechanisms. So like in case of lac operon, we understood that there is one repressor protein, which is keeping my lac operon off in the absence of lactose. And TRP operon is an example of TRP operon is an example of. So here also a repressor protein is produced, but it is negative repressible. Why it is negative repressible? Because in normal conditions, my TRP operon will remain switched on. It will remain switched on, and it will only remain switched off when tryptophan is present. So it is an example of negative repressible because here an induced or here a signaling molecule has to act to turn off the operon. So that is why it is negative repressible. Okay. So here the repressor protein which is produced, that is inactive. That is inactive. Here an inactive repressor protein is produced. So because it is inactive, it cannot bind to the it cannot bind go and it cannot go and bind to the operator region. It will remain inactive. So it will need a co-repressor. Here the co-repressor is tryptophan. So only when it binds to the tryptophan, only when this repressor binds to the tryptophan, it will become active. Now this is active. So after this repressor binds to the tryptophan, it becomes active, and now it can go and bind to the operator region and stop the or turn the operon off. Okay. So that is that is why it is called repression. So this is repression, and what is attenuation? So we will come to attenuation. So let's understand what is repression. So here you see that this is my regulatory genes, which is the TRPR gene. So that was in case of lac operon, it was lac-I gene. So this figure has been taken from the book Benjamin Pierce. So he if you this is a very good figure. So I've taken. So this TRPR gene, it is under the control of this of its own promoter. Regulated gene has its own promoter. So you see here that this repressor gene is getting transcribed and translated, and it is producing a repressor protein. Now this repressor protein is inactive. So it is inactive. So now since it is inactive, it is not able to bind to the operator region, and my operon is remaining switched on. It is remaining switched on, and all the genes, different E, D, C, B, A, these genes are getting transcribed, and the proteins are getting synthesized. Okay, so this is the condition when my tryptophan is low. Now, when tryptophan is high, this tryptophan goes and binds to the repressor. So this tryptophan is acting as a co-repressor. It is acting as a co-repressor, and when this tryptophan, so you see here now this tryptophan, once this tryptophan has bind to the inactive repressor, it has made this repressor protein active. Now, once this repressor protein is active, it will go and bind to the operator and prevent the RNA polymerase from transcribing the downstream genes. So that is what the TRP repressor binds to the operator and shuts the transcription off. So that is why it is a negative repressible system. So because negative why? Because here a repressor is getting synthesized, and repressible why? Because here a molecule is required for turning off the operon. Now, what is attenuation? So you need to understand a very important concept, a basic concept of transcription that is different between prokaryotes and eukaryotes is that the transcription in prokaryotes is coupled to translation. Okay, because it is taking place in the cytosol. So what happens is when the tryptophan level is low, the RNA polymerase begins transcribing the DNA producing regions one of the 5' untranslated region. So ahead or upstream of the promoter region, there is another region in the DNA, which is labeled as 1, 2, 3, and 4. So when these regions are start getting transcribed, so here you see that this is my RNA polymerase. This RNA polymerase is transcribing the gene, right? And transcribing the gene, and you can see here that the mRNA molecule, my mRNA molecule is getting transcribed or getting synthesized. Now, you can see in this figure that both the processes are taking place simultaneously. The RNA polymerase is transcribing the mRNA, and at the same time, the ribosome, my ribosome has attached to the mRNA molecule, and it has started translation of the mRNA to produce protein, to synthesize my protein molecule. So, now, that is what it has been explained here. A ribosome attaches to the 5' end. So this is the 5' end, and this is the 3' end of the RNA. Okay, so the ribosome has attached to the 5' end of the untranslated region. So this is actually when you learn about translation, you will see that if you that there are different structures in the mRNA that does not get translated. So these are called the 5' untranslated region and the 3' untranslated region. So at this 5' untranslated region, the ribosome attaches and translates region while region 1 while our region 2 is being transcribed. So the region 2 is getting transcribed. My region 1 has already been transcribed, and the region 1 has started translating is being translated by ribosome into protein. The ribosome stores at the TRP codon. So there are TRP codons in this region 1. So it will stall at the TRP codon. Why? Because my media has low tryptophan. So since the tryptophan level is low, the tRNA, when you will learn about this translation, you will understand about the tRNA. There are anticodon sequences which this anticodon will actually tell which amino acid should be or which is the next amino acid that will be joined to the growing polypeptide sequence. Okay. So because the ribosome is stalled, region 2 is not covered by the ribosome when region 3 is transcribed. Okay, region 2 is not covered by ribosome. When region 3 is transcribed, it pairs with the, so you see here that the RNA polymerase has stalled at this region, at the region 1. I stalled at the region 1. Why? Because here my tryptophan level is low, so it is not able to move ahead. Because the tryptophan is levels are low, it cannot attach the tryptophan to the growing polynucleotide chain, and it has stopped at this position. Now, but what is happening by RNA polymerase is moving ahead, and it is transcribing the region 2, 3, and 4. Now, once the region 4 is transcribed, this region 2 and 3, they are complementary to each other, and a hairpin-like structure is formed. Now, once this hairpin-like structure is formed, it is so because region 3 is already paired with region 2, the attenuator never forms and the transcription continues. So you have to understand that when this 2, 3 hairpin structure is formed, it will enhance or it will, but the transcription will not stop. The transcription will not stop. While when the 3 and 4, when the hairpin structure is formed between region 3 and 4, only then my transcription will stop. Okay, so that is what is explained here. So when tryptophan level is high, so when tryptophan level is low, you would want the tryptophan operon to stay on. You would want the tryptophan operon to stay on. Okay, while when tryptophan level is high, what will happen? Tryptophan level is high. This RNA polymerase is transcribing the mRNA. So region 1 here. So here, these are the TRP codon. So the TRP is also available. So the RNA polymerase will not stall at region 1. It will move ahead. It will move ahead to region 2. It will move ahead to region 2 now, but at the same time, the RNA polymerase is also transcribing region 3 and region 4. Now, since the region 3 and region 4 are complementary to each other, when this structure forms, when this hairpin structure is formed between region 3 and 4, it acts as an anti-terminator. This is my anti-terminator structure. So why it is called anti-terminator? Because it prevents it terminates transcription. So the ribosome covered parts of, so you have to understand the ribosome covers part of region 2. Ribosome is now covering part of region 2, preventing it from pairing with region 3. So now, since this ribosome, you see here, ribosome has come to region 2 and it is not allowing the region 2 to form a complementary structure or complementary bond with the region 3. Okay, so at the same time, the region 4 has also been transcribed. So now this region 3 and 4 come together and form a complementary structure, and thus producing the attenuator. So the attenuator is this structure. This is the attenuator 3 and 4. Okay, so this is why it is called that it is regulated by both repression and attenuation. Okay. So this is the structure of tryptophan operon. So this tryptophan operon it consists of tryptophan E, D, C, B, A. Okay, and there are 5 enzymes involved. There were only 3 enzymes which were involved in the breakdown of lactose into glucose and galactose, and subsequently uptake of this lactose into the E. coli cell. So here there are 5 enzymes involved. The controlling site in the TRP operon lies next to TRPE. So next to TRPE, there is a site which is called the TRPL. So this is the leader sequence actually. Okay. So this leader sequence will code for a leader peptide. Leader peptide. Okay. This leader region TRPL codes for a leader peptide and an RNA attenuator. So that is so that I was explaining here. That is the RNA attenuator, and this leader peptide is involved in the attenuation mechanism. So the TRPL codes for leader peptide and an RNA attenuator. It also contains a repressor regulatory gene called TRPR. So this TRP operon, it consists of a TR regulator gene. There is a leader sequence. This leader sequence is covering for a leader peptide and an RNA attenuator, which prevents the transcription, binds to the operator, blocking transcription of the TRP operon by inhibiting the RNA polymerase binding. So the next question is the leader sequence of the TRP operon is a stretch of nucleotides between. So that you just now I saw you I explained the figure as you saw in the figure that this leader sequence is present between the it is present between the you see the figure again. This leader sequence is present between the operator and the structural gene. Okay, it is present with the operator and the structural gene. So the answer is option A. Okay, so it is possible to obtain more strict regulation in E. coli by repressing transcription initiation alone. So here, what is happening by attenuation mechanism? What it is doing? It is allowing it to conserve its energy by inhibiting at the transcription initiation state. So the transcription is not allowed to initiate at the very first place. It is inhibiting the process. Okay, more strictly by replacing transcription initiation alone, but translation mediated transcriptional attenuation offers additional regulation. The attenuation site in the TRP operon is situated after the transcription start point. Most transcription stops here where when tryptophan levels are high. So when the tryptophan levels will be high, you saw that this ribosome has moved forward and it is now binding to the region 2 of the mRNA, and now 3 and 4 will form an RNA attenuator and prevent the transcription. Okay. Transcription continue to produce the functional TRP mRNA. The leader sequence stretch of nucleotide between operated and structural genes. So you have to, so this is what has been asked in the question as well. It is a stretch of nucleotide between the operated and the structural gene. Tryptophan concentration, when it is low, the entire operon, including the leader sequence, is transcribed into mRNA. So the leader sequence as well as this entire structural gene mRNA will also be transcribed. But when the tryptophan concentration is high, only 140 nucleotides, which is part of the leader sequence that precedes the attenuator, are transcribed into mRNA. The structural genes are not transcribed. So when the tryptophan concentration is high, the structural genes will not be transcribed. Okay. These will not be transcribed, but this TRPL, there will be this 140. That is what is explained as said over here, that only 140 nucleotides, which is part of the leader sequence that precedes the attenuated, only that portion will be transcribed. And that portion is enough to inhibit the transcription of further mRNA. Which of the following acts as a regulatory gene of arabinose operon? So this arabinose operon has also been discussed in the last lecture of week 6. So this arabinose operon is controlled by the ARA-C. Okay, you can go through the lecture. It has been explained very nicely. So we remember it is ARA-C. So we will just go through this ARA operon in brief. So negative. So here it is. There are three structural genes BAD. So there it was EDAB. EDCAB, here it is, BAD. So there is a regulator protein RSC here. So the cells do not require the ARA-BAD products to metabolize arabinose when it is not present. So this is actually similar to lac operon. Okay. So because in lac operon also what was happening when lactose was not present, the lac operon was remaining turned off. Okay. Lac operon was turned off. Here also when arabinose is not present, then the operon will remain turned off. So what happens? The dimeric RSC functions as a repressor. Okay, how does it function as a repressor? So you see here that RSC is actually a can form dimer. It can form a dimer, and one monomer binds to the ARA-BAD's gene operator. ARA-O2, and the other monomer binds to the remote DNA half site, so it forms a loop-like structure. So once this loop-like structure is formed, the RNA polymerase cannot bind to this promoter region, and transcription of these genes will remain turned off. Okay, a DNA loop is created as a result. The ARA-BAD promoter cannot be bound by as a result, cellular is blocked. Okay, well, positive regulation, how positive regulation takes place? Now this RSC, this is again the inducer here. So this is negative regulation, negative regulation of L-arabinose operon via ARA-C protein. So ARA-C protein is negatively regulating the arabinose operon. Why? Because it is keeping the operon turned off. Now when arabinose is present, it is binding to the ARA-C protein, it is causing a conformational change, and as a result, the RABD operon gets activated for expression, and ARA-C and CAP cooperate and act as activator when arabinose is present. So when glucose is absent, a high level of CAP protein or CAMP complex bind to the CAP binding site. Binding of CAP-CAMP is responsible for opening up the DNA loop. So there is also a cyclic AMP CAP binding site in ARA operon as well. Just as we had seen in lac operon, there is a CAP site binding site in ARA operon as well, increasing the binding affinity of ARA-C protein for ARA-I1, and thereby promoting RNA polymerase to bind to ARA-BAD promoter to switch on the expression of the ARA-BAD required for L-arabinose metabolism. So positive regulation of L-arabinose operon via RSC protein. So this is again one of the slides from your lecture that was covered. So arabinose operon regulation. You see, this is the a cartoon structure of the C protein. So you can see that it has a DNA binding domain and it has a dimerization domain. Okay. So in addition to being under the control of CAP-CAMP activator, the L-arabinose system is also positively or regulated, negatively regulated by the binding of the RSC protein. So RSC protein has the ability to act as both as an activator and a repressor. So, it can both negatively and positively regulate the ARA operon. So it forms a homodimer and interacts with the operator and initiator regions of the L-arabinose operon to control the transcription of ARABD. A DNA binding domain and a dimerization domain make up each RSC's monomer two domains. The binding of the arabinose is carried out by the dimerization domain. Upon the binding of the arabinose, RSC undergoes a conformational shift and adopts two different conformation. The binding of the allosteric inducer arabinose. So once the arabinose binds to the this dimerization domain, there is a conformational change which causes the RSC concentration to rise, and it can now adversely auto-regulate its own expression by attaching dimer to the operator region. Okay. So there are three structural genes, ARA, B, A, D, and what do they code for? You have to remember this. So B codes for ribonucleokinase, A codes for isomerase, and D codes for epimerase. So the correct answer is ARA B. A codes for what? It codes for isomerase and not epimerase. D codes for epimerase. Okay. Transacetylase is not there in araoperon. It is in lacoperon. Okay. So it is composed of three structural genes: ARA B, ARA A, ARA D, and there are operator regions. There are two operator regions: ARA O1, ARA O2, and there is an initiator region: ARA I1, ARA I2. So this is ARA I1 and ARA I2. So this is the structure of the ARA operon. Okay, the structural genes ARABAD encode enzymes for L-arabinose catabolism. So breakdown of the arabinose. So that is arabinose catabolism. And there is also a cap binding site. You see here, there is a cap binding site between the ARA O1. So there are two operator sites, there are two initiator sites, there is a cap binding site. Okay. Yeah, so this when this cap binding site, what will happen? The cap-cam complex will bind and facilitate catabolite repression and result in positive regulation of the ARABAD when the cell lacks glucose. Positive regulation, when will the positive regulation of ARABAD take place? When the cell will lack glucose. The regulator gene RSC is located upstream of the L-arabinose operon. So this is the regulatory. Where is the RSC? This is the regulatory gene. It is located upstream. You can see, as you can see in this figure, it is located upstream of the promoter region. Both RSC and ARABAD. So RSC has its own promoter. This is the promoter for RSC. PPARSC, this is the promoter for RSC gene, and this is the promoter for the BAD gene. So there are separate proteins, so that is why it is a regulatory gene. So regulatory gene has its own promoter. Regulatory gene is not a part of an operon. Both RSC and RBAD have and RBAD and RSC are transcribed in opposite direction. So you see here that the transcription of this BAD gene will take place in this direction, while the transcription for this RSC protein takes place in the opposite direction. So that transcription of the RSC protein or the RSC mRNA and this BAD mRNA are in opposite direction. Okay, so these are some points you have to remember. You have to remember the proteins which are synthesized from these structural genes. From B it is ribonucleokinase, A it is isomerase, and D it is epimerase. So that is all for today's session. So So I hope this session proved to be useful and you would understand some of the important concepts that were covered in week 6. So if you have any more doubts, you can take it up in the next class. So thank you for your patient listening. Bye.